Alright, Mr. Best, on this side of the paper, this is the light reaction that happens in the chloroplast. So, as electrons pass through the carrier of the redox reaction, the hydrogen ions are moved from the stroma and are deposited in the thylakoid space, which is right here. So from here, which is the low concentration of hydrogen ions, will go to the high concentration of hydrogen ions, which is the thylakoid space. And then it will be storing energy. It will be storing energy as a proton motive force, which is the hydrogen gradient. And the water right here is splits into the photosystem 2 on the side of the membrane facing the thylakoid space. And as platosquanine, or PQ, which is this, it will transfer the electrons to the cytochrome complex. And then the protons are translocated across the membrane into the thylakoid space. And the hydrogen ion is then removed from the stroma, and it will be taken up by NADP plus. Sorry. The diffusion of the hydrogen ions from right here, they'll be going through the ATP synthase. It will help power it up. So basically, the NADPH and the ATP will help power up the Calvin cycle, which is right here. And in the Calvin cycle, for every 3 molecules of carbon, which is the starter, every 3 molecules of carbon that enters the cycle, the net output is 1 molecule of G3P. The light reactions will help sustain the Calvin cycle by generating ATP and NADPH. So right here, as the carbon enters the cycle, Rubisco will join it. It will make 3 short-lived intermediates, and then it'll go through 3 phosphoglycerate. It makes 6 of them, and then that's where ATP will then turn into ADP, and then it'll make one biphosphoglycerate, which will go from NADPH, and then it'll go to NADP plus, and then we'll get a phosphate group out of it. And then it'll go to glyceraldehyde, 3-phosphate, and then out of that it will come out and make a G3P, which is a sugar, and then it'll make 5 more G3P, and then which ATP is used, and then it'll go back to ADP, and then it'll make the RUBP, and the whole cycle starts all over again. I think it has to go through 6 times in order for it to make 1 glucose molecule. And then up here is where the C4 plants and the CAM plants. So in the C4 plants, the carbon fixation and the Calvin cycle occur in different types of cells. So this is the mesophyll cell, which is between the bundle safe and the leaf surface and that's how it's arranged. And for the CAM plant, the carbon fixation and the Calvin cycle occur in the same cell at different times. So it happens in night and day. So basically, it's like the mesophyll cells. In the mesophyll cells, the enzyme PEP carbolase adds carbon dioxide to PEP. And then a 4-carbon compound conveys the atoms of the carbon dioxide into the bundle shaped cell via plasmodesmata, the junction in plants. And then lastly, the bundle shaped cells, the carbon will be released and it will enter the Calvin cycle, which will go right back to here. And that's pretty much it for my project. I'll give it to you tomorrow. Alright.